So ELiPSE-1 is a phase 1 dose escalation study of a CD19 CAR-NK that's iPSC-derived. The CAR-NK cell has multiple CRISPR-based edits to namely avoid T-cell mediated rejection from the host and host NK cell mediated. And so the study is currently ongoing, enrolling patients with relapsed refractory B-cell non-Hodgkin's lymphomas. Patients in the study have to have had 2 prior lines of therapy and can have had a prior line with CD19 targeting therapies. So at the ASCO meeting, we presented safety data on the first 12 patients enrolled in the study. These patients were enrolled across 2 different dosing schedules, one in which a single infusion of CNTY-101 was given after standard LD, and then a second schedule in which CNTY-101 was given on day 1, 8, and 15 after LD chemo. And in both schedules, patients could receive additional cycles of treatment with or without LD chemotherapy after at least 28 days from the first cycle, and if they'd had stable disease or better response. And so we updated safety data for the study. The majority of patients enrolled so far had diffuse large B-cell lymphoma. About 25% of the patients had had prior autologous CAR T-cells. And overall, the safety data at an early point looks promising. Most of the reported adverse events are due to the LD chemotherapy. Cytokine release syndrome was fortunately uncommon, seen in 33% of the patients, mostly low grade and short duration. And ICANS was uncommon, seen in just 1 patient, and was also low grade. And I think excitingly, we have some initial efficacy data as well. The overall response rate for 10 patients that were efficacy evaluable was 40%, with a 30% complete response rate. And then we also presented some correlative data that demonstrated these iPSC-derived CAR-NK cells are detectable and persistent in extravascular spaces. And in the very first patient enrolled in the study who had had 7 total cycles of therapy, including 5 without LD, there was no evidence of allorejection of the CAR-NK cells. So the study's continuing in dose escalation in both schedules, and we look forward to hopefully updating safety and efficacy data in the near future.